Essentials in cell biology. Learning objectives. Understand the difference between two major categories of cells, prokaryotes and eukaryotes. Highlight the inner structure of the eukaryotic cells. Underline the process of protein expression in eukaryotes. In all living systems, there are two main categories of cells: prokaryotic cells and eukaryotic cells. Prokaryotic cells are much simpler in structure compared to cells in the other group. They lack a membrane-bound nucleus. Instead, the DNA in prokaryotic cells is typically located in the cytoplasm. Eukaryotic cells are typically found in multicellular organisms like plants, animals, and fungi. They have a well-defined nucleus that houses the cell's DNA. In terms of size, prokaryote cells typically range from about 0.5 to 5 micrometers in diameter, while typical animal and plant cells are about 10 to 30 micrometers in diameter. Let's focus on the content of a bacterium. In all bacterial cells, you will find plasma membranes. Many of these cells also have a cell wall, while in some, there is an additional capsule. The primary DNA of the cell is situated within a region of the cytoplasm known as the nucleoid. Ribosomes are also found within the cytoplasm. Hence, in bacterial cells without membrane-bound nuclei, in the same location, genes can be transcribed and translated. Plasmids are additional strands of DNA that may carry extra genes. They are not essential for the basic survival of the cells. Unlike bacterial cells, eukaryotic cells are complex and compartmentalized, with various regions within the cell, separated by membranes. In addition to a plasma membrane, there are mitochondria functioning as the cell's energy generators. The cytoskeleton acts as the structural framework that is responsible for maintaining the cell's shape and integrity. The nucleus is the crucial site housing the cell's primary DNA. It is the place where genes are, and where the transcription takes place. Taking a closer look, chromatin, comprising DNA and associated proteins, and nucleoplasm are enclosed by a nuclear envelope. The envelope consists of two membranes and includes various nuclear pores. The nuclear envelope has a close relationship with the endoplasmic reticulum, ER. As we will explore later, the ER's function is interconnected with that of the Golgi apparatus. As expected, in eukaryotic cells, functional ribosomes are found in the cytoplasm, where translation occurs. In eukaryotes, after RNA is produced in the nucleoplasm, it must pass through the nuclear pore, and translation takes place in a distinct cellular compartment. While in bacteria, transcription and translation can simultaneously occur in the same location. There are also some minor differences in the ribosome size and structure between bacteria and eukaryotic cells. There is also a difference in codon preference between bacteria and eukaryotes. Let's look at the translation process of an mRNA. In both bacteria and eukaryotic cells, the ribosome subunits come together on the mRNA to initiate translation at the start codon and continue until they reach the stop codon. After completing the translation, they separate once again. In eukaryotes, proteins destined for most organelles, the nucleus, and the cytoskeleton are synthesized through this pathway. However, there is an alternative pathway for producing other proteins. Let's dive into the cytoplasm of a eukaryotic cell to explore the alternative pathway. In this pathway, after the ribosome subunits join and translation of the mRNA begins, a small segment of the polypeptide known as the signal peptide is generated. This small peptide is recognized by a signal peptide recognition protein, causing translation to temporarily stop. Translation resumes shortly after the complex attaches to the ER. Upon resuming translation, the polypeptide passes through the membrane on the ER and enters the lumen. This is the point at which the signal peptide can be cleaved from the polypeptide being produced, catalyzed by an enzyme known as signal peptidase. Upon reaching the ribosome at the stop codon, translation terminates, leading to the separation of the two ribosome subunits, and the mRNA is then free to degrade. Now let's look at the final part of this pathway from another point of view. Inside the ER, the produced proteins undergo various modifications. It prepares them for their ultimate structure and their functional role. These proteins are transported inside the cytoplasm via small vesicles. On their way, they land on the Golgi apparatus. As they traverse the Golgi systems, proteins undergo further modification. Finally, depending on their intended location and function, these proteins can be directed to either the cell membrane, lysosomes, or secreted outside of the cell. In this video, we explored the distinctions between prokaryotes and eukaryotes, considering factors such as size, structure, and the flow of information. We delved into the inner structures of eukaryotic cells. We emphasized the pathways for protein expression in eukaryotic cells.